All right, everyone, let's get started. Today, we're diving into a really clever paper from Zaman Wu and his team at NUS and SJTU. They're tackling a fundamental problem in our field with an elegant analogy, LLM DNA. It's a powerful idea for tracing the evolution of these models, and I think you'll find their approach quite compelling.
So the motivation here is something I'm sure we've all run into, the Hugging Face ecosystem. It's a jungle, right? There are literally millions of models, and their lineage is often a complete black box. So the authors are asking a simple but really profound question. How can we start to systematically map these relationships?
And this TSNE plot from the paper, well, it just drives the point home. Each dot here is a model, and you can just see the chaos. I mean, sure, we can see these vague families of models, but the connections are messy, they're often undocumented. What we really need is a principled way to draw a map of this entire space. Okay, so why is this more than just an academic exercise in tidiness? The authors highlight three really critical applications. First, safety and security. Think about tracking the propagation of vulnerabilities like backdoors as they get passed down through fine-tuning. Second, model governance. How on earth do we enforce licensing if we can't even tell where a model came from? And third, something I know some of you are working on, in multi-agent systems, understanding the functional diversity of our agents is absolutely crucial for effective design. So, with the problem clearly established, the paper moves into its core theoretical contribution, and that is to formally define and then prove the concept of LLM DNA. The whole idea is built on this beautiful analogy to biology, right? Just as the discovery of DNA gave us a framework to understand the relationships between organisms, the authors are proposing an LLM DNA to map the relationships between models. It's a really powerful way to frame the problem. So what is this LLM DNA? Well, before we get into the weeds with the math, the intuitive idea is pretty straightforward. It's a compact, low-dimensional representation of a model's functional behavior. Think of it as a small vector, a little fingerprint, that captures the essence of what the model actually does. Now, to make this rigorous, they start by defining an LLM as a function f that maps text to logits. But the critical move here, as you can see in definition 3.1, is proving that the space of all these functions, f, is a Hilbert space. This is a crucial theoretical step because it gives them a space with a well-defined notion of distance and geometry, which is absolutely essential for the entire framework to work. Okay, with the Hilbert space established, they can now give us a formal definition for the DNA. As you see in definition 3.3, it's a low-dimensional vector that you get through a bi-Lipschitz map from that functional space. Now, the key property of a bi-Lipschitz map, and this is the whole point, is that it preserves the metric structure of the space. In plain English, it means the distance between any two models in that original crazy high-dimensional space is preserved up to a constant factor in their low-dimensional DNA space. This is the mathematical guarantee that functionally similar models will have nearby DNA. And from this single bi-Lipschitz definition, two key properties just naturally emerge. It's really neat. First, you get inheritance. Because the map is continuous, small changes in the function space, like fine-tuning a model, have to lead to small changes in its DNA. Makes sense. Second, you get genetic determinism. Because the map preserves distances, models with similar DNA must have been close in the original function space, which means they have to have similar behavior. It's a very elegant theoretical result. So the theory is elegant, but does a mapping like this actually exist in practice? This is where the Johnson-Linden-Strauss lemma comes to the rescue. It's a classic, beautiful result that tells us, yes, we can use a simple random projection to map high-dimensional data down to a lower-dimensional space while preserving all those pairwise distances. So this not only proves that their LLM DNA exists, but it also gives them a practical recipe for how to actually extract it. So with the theory laid out by the JL lemma, let's look at how they actually go about implementing this. This brings us to their DNA extraction pipeline. And the resulting algorithm is just beautifully simple, and maybe most importantly, it's training free. You just probe the model with a set of prompts, you embed its responses, you concatenate them all into one big high-dimensional vector, and then you just hit it with a random projection matrix. And that's it. It's an incredibly pragmatic approach that flows directly from the theory. This diagram from the paper gives a great visual overview of that entire workflow we just walked through. It's a clean, straightforward pipeline from prompts all the way to the final DNA vector. Okay, so the theory is sound, and the pipeline seems really practical. The next logical step, of course, is to put this thing to the test. The authors ran their experiments on a large set of 305 real-world models to see how it actually performs. First things first, they needed to make sure the method is stable. Does the DNA you extract depend heavily on the specific prompts you use? This mantle test shows the answer is no. They calculated the DNA distances using two totally different data sets and found a very strong Pearson correlation of 0.78. This tells us the underlying geometric structure of the DNA space is consistent and robust. So, does it work? Well, this table says yes, resoundingly. Using Hugging Face's own metadata as the ground truth, their DNA method hits an F1 score of 0.88 and an AUC of nearly 0.96. That's a massive improvement over both the random and greedy baselines. It tells us this is a highly effective way to identify real model relationships. And here's that TSNE plot again, but now we can really appreciate what we're looking at. Each one of these points is a model's DNA, and you can see these distinct species forming. The llama family is clustered over here, the Qin family is over there. The clear separation between these clusters is just a powerful visual confirmation that the DNA is capturing really meaningful functional relationships.
But what's even more powerful is when the DNA uncovers things that aren't documented. They highlight this great example of Vicuna 7b v1.1. Its model card says it was fine-tuned from llama, but it doesn't specify which version. The DNA plot, however, places it squarely inside the llama base cluster, which strongly suggests its specific parentage. It's basically a paternity test for LLMs.
And this leads to the ultimate payoff of this whole DNA analogy. Can we use it to reconstruct the evolutionary history of the entire field? Can we actually build the LLM phylogenetic tree? So they take the analogy to its logical conclusion. They ask, if we have their DNA, can we use standard phylogenetic methods from biology to build an actual evolutionary tree of LLMs? And this is the result. This is, as far as I know, the first ever phylogenetic tree of LLMs built purely from functional data. No metadata, no release dates, just the behavior of the models themselves. It's a fascinating look at the evolutionary history of our field, reconstructed from the ground up. And you can pull some really interesting insights just from looking at this tree. First, you can see the major architectural shift, from the older encoder-decoder models like FLAN T5 at the root, to all the modern decoder-only models branching off. The structure also broadly aligns with the temporal progression of releases from 2023 to 2025. And finally, the branch lengths themselves might suggest different evolutionary speeds. Look at the longer branches for the Quen and Gemma families. It suggests a faster functional evolution compared to the llama series. So, to conclude, this paper gives us a powerful new tool, a new lens, for understanding the LLM ecosystem. But the authors leave us with a really tantalizing thought for the future. We now have a way to read LLM DNA. The next logical step, then, is to figure out how to edit it. Can you imagine targeted, functional modifications at the DNA level? It's a really exciting direction for future research. I'll stop there and take any questions.